Now, my name is Dr. Mariel Pikkemaat and I'm the coordinator of the GPI AMR project REBEL, which is short for Repotentiating Beta-lactam antibiotics. Now, in this webinar, I will try to explain a bit more on what we do in this project, but first of all, why we do this. Now, the success of antibiotic therapy is seriously threatened by the increasing prevalence of antimicrobial resistance. And AMR actually threatens many of the most important medical advances we've made. The UK Independent Review on Antimicrobial Resistance, which was initiated by the UK government and the Wellcome Trust, published a first report in December 2014 called Antimicrobial Resistance: Tackling a Crisis for the Future Health and Wealth of Nations. Now, in this report, they demonstrated there will be a profound health and economic consequences for the world, especially in emerging economies, if antimicrobial resistance is not tackled. They estimated that drug-resistant infections already cause up to 50,000 lives lost each year in Europe and the US alone, and 700,000 worldwide. But if we don't take action, this might rise to 10 million in 2050. Now, to avert this crisis, there is not a single solution. We need to create awareness, stimulate consumer education, and make people understand that antibiotics are not a cure for a common cold or flu. We should optimize treatment with precise and fast diagnostics, limiting the use of broad-spectrum and last resort antibiotic drugs. We should stop using antibiotics as growth promoters in animal production. Now, fortunately, the EU has realized this practice is highly undesirable and banned the use of antimicrobial growth promoters. But it has been estimated that, for example, in the United States, more than 70% of all antibiotics are fed to livestock, creating a massive opportunity for the development of antimicrobial resistance. Now, any use of antimicrobials, even when they are used appropriate and conservative, contributes to the development of resistance. So we will need to develop new antibiotics. However, the short-term perspective with respect to the clinical introduction of new antibiotics is alarming, since the output of pharmaceutical industry has shrunk to an absolute minimum. Research efforts in this area very much focus on identifying new microbial targets for antibiotic therapy, which is essential, but also a laborious and lengthy strategy. Alternatively, restoring the sensitivity of resistant bacteria to existing antibiotics could provide a more expeditious approach. Now, in this presentation, I'd like to show how with this project we are contributing to the development of new drugs, or rather the restoration of activity of a proven successful drug, the beta-lactam antibiotics. Now, the project is a collaboration between Radboud Wageningen University and Research Center from the Netherlands, which is responsible for the development of the bioassays and the isolation and identification of compounds, the University of British Columbia in Vancouver, Canada, with the structural biology group of Natalie Strinetka, who holds a large expertise in beta-lactamases, and the organic chemistry laboratory of the Umeå laboratory in Sweden of Professor Fredrik Almqvist, who has great expertise in the design and synthesis of novel antibiotic agents. Now, beta-lactam antibiotics are among the most successful and widely used drugs for treating bacterial infections. They have a long history, starting from the discovery of penicillin by Alexander Fleming in 1928. Penicillin was first introduced in clinical practice in the 1940s, and its success triggered a major search for other antimicrobial compounds, both naturally occurring and semi-synthetic derivatives. Now, the currently available beta-lactam antibiotics encompass penicillins, cephalosporins, carbapenems, but also cefamycins and monobactams. But the first three are the most important. They are all structurally related through the presence of a beta-lactam ring in their molecular structure, while the two-ring system, also called the nucleus, differs between these three basic molecules. Now, many semi-synthetic derivatives of these compounds have been developed, which further expanded the activity spectrum of the beta-lactam agents. Now let's look a bit more in detail into the antibacterial mechanism of these beta-lactam antibiotics. Basically, the bactericidal effects of beta-lactam antibiotics are achieved through the inhibition of the bacterial cell wall synthesis. The bacterial cell wall essentially consists of long chains of peptidoglycan, which is a polysaccharide, that are cross-linked through peptide bonds. Now, the cross-linking of these peptides is done by bacterial enzymes called transpeptidases, and it's this transpeptidation reaction that is inhibited by the beta-lactam antibiotics. Now, the beta-lactam antibiotics act through the covalent modification of these transpeptidases, inactivating them and thus stalling the cell wall assembly in bacteria. Now, inhibiting this essential step in the bacterial cell wall synthesis ultimately results in osmotic lysis and death of the bacterial cell. Now, the same Alexander Fleming that discovered this miracle drug also already predicted the problem of resistance development as an inevitable evolutionary consequence. The most common form of beta-lactam resistance is the production of beta-lactam hydrolyzing enzymes, so-called beta-lactamases. Now, bacteria have evolved enzymes capable of cleaving the essential beta-lactam ring, which leaves a molecule that is no longer capable of binding and inactivating the bacterial transpeptidase enzymes, resulting in a beta-lactam resistant bacterium. Now, the number of unique naturally occurring beta-lactamases now probably exceeds 1,300 and is rapidly growing. There are two main molecular strategies employed by these beta-lactamases to hydrolytically cleave the beta-lactam ring of penicillins and cephalosporins, either through the action of an active site serine nucleophile or through the activation of a water via a zinc center, and then it's a so-called metallobeta-lactamase. Now, these serine beta-lactamases and metallobeta-lactamases can be further subdivided based on their three-dimensional structures, or the amino acid sequences, which is generally referred to as Ambler classification. It's, however, also common to group them according to substrate preferences and inhibitor sensitivities, which is actually a functional or a biochemical classification as it was proposed by Bush, Jacobi and Medeiros. Now, the term extended spectrum beta-lactamase, or ESBL, applies to beta-lactamases capable of hydrolyzing so-called third and fourth generation cephalosporin, such as cefotaxime and ceftazidime, which have an expanded antibacterial spectrum. Now, the metallobeta-lactamases all share the property that they can hydrolyze carbapenems, which however is not a biochemical characteristic that is exclusively limited to the metallobeta-lactamases. From this scheme, it can be concluded there are several beta-lactamase groups that can hydrolyze virtually any beta-lactam antibiotic. Now, the next few slides will show us the speed at which the spread of these ESBLs is currently going on. This picture shows the prevalence of ESBLs represented by clinical E. coli isolates in different European countries in the year 2005, so about 10 years ago. They have been collected by the European Centre for Disease Prevention and Control, and they can be accessed from their antimicrobial resistance interactive database, ESNet. Now, as you can see, there is considerable variation in ESBL prevalence in Europe. This variation is to a large extent linked to the differences in how heavily they use antimicrobial drugs. Now, in 2005, the majority of the EU countries are still shown in green, meaning that the ESBL prevalence is below 5%. Now, the next slide shows the situation in 2010, and it shows the majority of the countries between 5 and 10%, with only the Scandinavian countries keeping a relatively low prevalence. Now, the next slide, however, shows that also these countries can't escape this ongoing evolutionary rat race, and we see a continuously increasing prevalence, with now a considerable number of countries reaching resistance ratios of over 25%. Now, it's not hard to imagine where this will lead to if we don't come up with solutions. One way of circumventing beta-lactamase resistance is through the co-administration with beta-lactamase inhibitors. Now, resistant bacteria will still produce the beta-lactamase enzymes, but since it's no longer functional, it will not be able to hydrolyze the beta-lactam antibiotic, which can still exert its effect on the transpeptidases again, and the bacteria will die. The combining of a beta-lactam with a beta-lactamase inhibitor is an established and extensively used therapeutic strategy, which started with the acceptance of augmentin, which is amoxicillin and clavulanic acid combination, targeting class A serine beta-lactamase drug resistance in the early 80s. Two more structurally related beta-lactamase inhibitors made it to clinical application, sulbactam and tazobactam. Notably, none of these inhibitors effectively targets the rapidly emerging plasmid-encoded class D serine beta-lactamases or the metallobeta-lactamases. And also resistance has been observed in the clinic. Now, from an evolutionary point of view, this does not come as a surprise, since clavulanic acid, sulbactam and tazobactam holds significant structural similarity with the beta-lactam antibiotics. Now, the metallobeta-lactamases have a different active site, which will probably require a different type of molecules to inhibit this kind of beta-lactamase. So far, research in this area has mainly focused on the screening of chemical libraries and molecular modeling studies. This has yielded several structurally diverse inhibitors, but most of them are not suitable for therapeutic application. The only exception is the recently approved compound avibactam, which is actually the only non-beta-lactam beta-lactamase inhibitor available so far. So obviously, there is an urgent need for new beta-lactamase inhibitors. Now, to expedite the search for new beta-lactamase inhibitors, we will perform a large-scale screening of botanical extracts. Now, plants are continuously challenged by a broad spectrum of microorganisms and have evolved a complex arsenal of chemical defense mechanisms. They produce a wealth of secondary metabolites and are therefore considered a versatile source for mining new bioactive molecules. Now, our starting point is the plant extract collection that is accomplished by the Kiel University in Schleswig-Holstein, and it's called Package. Now, this library contains more than 4,600 standardized, unique, aqueous, ethanolic and other extracts from over 860 different plant species, covering over 190 plant families and 11 different plant tissues. They originate from health shops, outdoor cultivated plants, but also from traditional herbal medicine. To enable the selection of beta-lactamase inhibitors with the widest possible spectrum, our screening strategy involves a series of bacterial tester strains, bacteria producing a particular beta-lactamase, covering the most important beta-lactamase groups. We've created these tester strains by simply cloning the beta-lactamase gene into a standard IPTG-inducible E. coli expression factor. Then we transform a laboratory E. coli strain with a factor, and the addition of the IPTG will result in beta-lactamase production. For our screening assay, we look at the growth of the tester bacteria. Now, the library is dispensed in 384 microwell plates containing only 3 microliters of the extract. The bacteria are diluted in a nutrient broth, which additionally contains IPTG for the induction of the expression plasmid, so the production of the beta-lactamase. Now, the broth also contains a redox indicator, which initially has a dark blue color, but when the bacteria grow, it changes to pink. Now, the screening strategy is based on the comparison of 2 identical copies of the plate, of which one is additionally supplemented with beta-lactam antibiotic. Now, the plates are left in the incubator for around 20 hours. Now, if the bacteria start to grow, the well will turn pink, and if the growth is inhibited, the well will stay blue. Now, most of the botanical extracts will not inhibit bacterial growth, so after the incubation, the majority of the wells will turn pink. There will, however, be extracts that cause bacterial growth inhibition. Now, if growth inhibition is occurring in both plates, it's a general antimicrobial effect. The ones we are interested in, however, are the extracts that only inhibit bacterial growth in the plates supplemented with beta-lactam, since in these extracts the antimicrobial activity of the beta-lactam has been restored, indicating the inhibition of the beta-lactamase. Now, extracts showing beta-lactamase inhibitory activity will be subjected to further isolation, identification and structural confirmation of the bioactive compound. Identification of the inhibitors will be achieved through bioactivity-guided fractionation. Botanical extracts will be subjected to multidimensional chromatography, allowing single peak isolation, which will subsequently be retested in the original bacterial growth inhibition bioassay. Bioactive molecules from fractions containing inhibitory activity will be identified using mass spectrometry combined with mass database searching and NMR. Based on these potentially interesting hits, we will continue. The compound properties will be evaluated in silico by multivariate methods. Compounds most suitable to synthesis will be chosen in order to prepare a small, focused library of analogues for more detailed structure-activity studies. Now, the ability of the inhibitors to reverse beta-lactam resistance will also be verified in relevant clinical isolates to ensure efficacy in a proper genetic background. The toxicity of the compounds will be assessed in a panel of cytotoxicity assays, targeting different toxicological endpoints to allow elimination of cytotoxic compounds. Promising compounds will proceed to biochemical characterization and X-ray crystallography, which will be used to probe structural and mechanistic details on the mode of serine beta-lactamase or metallobeta-lactamase inhibitory action. These data will further provide essential insights, facilitating the design and optimization of downstream leads. This should ultimately result into the design of universal scaffolds with the potential to block simultaneously the serine beta-lactamases and metallobeta-lactamase variants. Now, this research has been made possible by the financial funding of the Swedish Research Council, the Canadian Institute of Health Research and the Dutch Organization for Health Research and Development. Thank you for your attention.